Hey everybody, Mr. Murphy here. I'm going to be covering 10.2, which is on transcription. This is the process in which RNA is created from a DNA template. And so the needed components are the DNA to read as a template, nucleoside triphosphates, which is your adenosine, guanine, cytosine, and uracil in a triphosphate format that's basically been prepared to be grabbed in the nucleus and turned into the RNA that's growing by Rna Polymerase, which is the enzyme that is responsible for it. So other than this, there's other types, or other than the mRNA that we typically see, there are other types of RNAs that are also produced, and that includes tRNAs, rRNAs. We'll see the small nuclear RNAs, which we call SNRPs, and then various other types of RNA products as well. So transcription is a way in which you turn DNA into RNA, and it turns out that RNA actually has a whole lot of useful functions inside of the nucleus and other places inside of the cell. In fact, a single-stranded RNA molecule acts a lot like a protein in terms of the folding that it does. We remember how proteins fold themselves in order to align their different amino acids that have different attractions or repulsions from each other. Well, an RNA strand is still going to have those hydrogen bonding sites on the A, U, G, and C, and so they'll fold over on themselves and match up as best they can just because of chemical forces, and those shapes can account for actions very similar to proteins. So we see a lot of functionality to the different types of RNAs that can be made. Throughout this process, RNA polymerase is sort of the star of the show. Much like DNA polymerase, it is responsible for the matching of a single-stranded DNA molecule that has been separated with its complementary RNA in this case. And so this can result in a single RNA polymerase rides along the template strand and produces potentially hundreds of RNA bases that are stitched together, and that would be the encoded mRNA or other type of RNA that's going to serve some function. They don't need primers like DNA does, but we'll see how there is still a control mechanism to sort of initiate when and how often this is occurring. And so here, this is sort of a kind of tough to wrap your head around, but we have the DNA double strand here, and then sort of within the structure of the RNA polymerase, that's where the separation occurs, and then the DNA would actually wind back up before it pops out the other side of this. So the exposed single-stranded sides of the DNA are kept within the confines of the RNA polymerase, which is important because of the importance of keeping your DNA safe so that you don't damage it, since you only have one set inside of the nucleus. So this happens in 3 steps. There's initiation, elongation, and termination. Initiation requires a promoter, and this is a special sequence in the DNA prior to the gene location that we want to transcribe, and this is where RNA polymerase is going to bind. Now, we'll talk about this down the road, but promoters can be sort of blocked or enhanced in their ability to attract the RNA polymerase, and that's part of the control mechanism that different organisms have to control how and when different types of transcription are occurring. This is also referred to as the transcription initiation site. Okay, and so here we have the double-stranded DNA. We're transcribing it into pre-mRNA. We'll turn that into mRNA, and then that's what ends up getting translated into a polypeptide. So initiation occurs. We have our RNA polymerase, which is bound at this promoter region. It then begins to move down the length of the DNA and unwinds it and begins to link these nucleic acids, the nucleotides, the RNA nucleotides, and that's the process of elongation. So this is where we are actually producing the RNA strand. So this is reading the DNA template in a 3 to 5 direction. I personally don't worry as much in terms of asking you questions about this as much as I do with DNA polymerase, because DNA polymerase and the whole leading and lagging strand thing has such importance on the directionality. It is also very important in terms of RNA polymerase. It's just a little bit less of a point of confusion. So when this is happening here, this is where these different ribonucleoside triphosphates are coming in and being added onto. So these are available nucleotides to be created into this growing RNA strand. Termination is then also a special sequence in the DNA which tells the RNA polymerase that you're done, it's time to stop, and that's when we terminate the process of creating the RNA. So after this, we have what is called pre-mRNA if we're transcribing mRNA. Now this may not always be mRNA, this might be creating a tRNA or some other type of RNA. So when we look at mRNA, there are different regions that are considered coding or non-coding. So non-coding regions, which are found in eukaryotic organisms, are introns. And these need to be removed. These are basically interspersed throughout the RNA strand and don't correlate to the codons and amino acids of the protein that ultimately we want to create. And so those will have to be ripped out, and we'll see how that happens in a second. The exons are the actual coding regions, so that's the part that we actually just want to keep. So those together are our pre-mRNA. And here you can see a single, this is the DNA here, and then we have our pre-mRNA here. And they had these little regions, exon, intron, exon, and we need to remove those introns. And so what happens is they physically get removed out of this molecule, and then just the exons are stitched together into our final mRNA, which can then be moved along to the cytoplasm where it can find ribosomes and do its job. There's a whole big process historically of how we identified this. They talked about this particular experiment where basically denature the DNA, probably using temperature, and then put a RNA probe into this, which is going to match up with the DNA here. And what can be revealed is that if there's just exons, then that mRNA is going to match up perfectly. But when there's an extra little bit in here, it ends up creating these little loops, and you can see here an electron micrograph of this showing this little loop. And so this kind of reveals the fact that the mRNA here does not match perfectly with the original DNA, that there was this section that ended up being removed. And the reason why it loops out is because this half of the mRNA is matching with this exon, and this half is matching with this exon, but nothing matches that middle bit. And so it sort of folds the DNA and creates this visible product. So introns interrupt, but they don't scramble the DNA sequence. So they're not like completely mixing up and causing any sort of like complete reformation of the DNA or anything like that, but they are these little interrupter sections, and they therefore need to be removed. And RNA goes through a process of splicing, which uses a different type of RNA called small nuclear ribonucleoprotein particles, which is we just call them SNRPs. And those are responsible for removing those and by creating something called a spliceosome. And this is pretty cool, and there's animations on YouTube and such that can show you how this happens. But basically, they bind to one side of the intron and the other, and then the two SNRPs here basically fold together. And they bring this thing into a loop, and we can rip off the top part of the loop and stitch together the two exons. And so then, and again, with the process of probably other proteins helping out with the actual binding of these things, and then we can obliterate that intron. Or that intron may serve a functional purpose. So introns, you may wonder, very good question, like why are they actually there? And there's actually a whole bunch of reasons that they could, in fact, be there. And that's a point of, I think, very active research in terms of the huge variety of types of introns that can exist. And there's experiments that you can read about that are kind of interesting, and where basically by altering the gene sequence to pre-remove those introns and then transcribing that segment, it leads to, or can lead to, diminished amounts of the actual final protein product, which suggests that the introns, these little pieces of RNA that are breaking off, may actually be very active in facilitating the success of the mRNA that is produced. There are different diseases that demonstrate, again, this is still looking at some historical components, diseases that demonstrate the association between mutations and the effects that they can have in the splicing process and in the successful production of the mRNA. There's also something called alternative splicing, which is a pretty mind-blowing thing where depending upon how the introns are removed or even which introns are removed, a single coding region of DNA could actually produce multiple different mRNA and therefore protein products depending upon the combination of regions that you leave together or remove as introns. And so you can kind of think of it as like if you're building some like Lego castle or something, and there's two towers and they're mostly identical, but one's got these little tweaks to it or something. That's kind of similar to what's going on here, where maybe we need to make a protein product it's a little bit different than the other one, rather than having two completely separate coding regions, which would be probably harder for that to have come into existence and evolved. Instead, we have a single one which can be sort of read or interpreted in two different ways depending upon the chemical makeup of what's going on around that protein during that transcription. So the concept of alternative splicing is pretty cool. In addition to the removal of these introns, there's two other components that need to be added to the pre-mRNA before it can be exited from the nucleus, and that is the 5' cap, or we call that the G cap. That's basically, it facilitates the binding of the mRNA to the nuclear pore to exit it out of the nucleus, and then also it prevents breakdown of enzymes. So think of it as like a cap, like it's a helmet on the end of that mRNA that makes it so that no enzymes or anything are going to be able to start ripping off pieces of nucleotides or anything, because that's the side of the RNA that is most important. That's the side that the ribosome is going to form around the start codon, and then if we started destroying that side of the RNA very quickly, it would be losing its function. The poly A tail, on the other hand, is on the other end. So this is after the stop codon. You have a whole bunch of A's, adenines, and this is basically, I think of it as like a fuse. When an RNA is out in the cytoplasm, there are a whole number of nuclease enzymes that are going to be breaking down any sort of nucleic acids that they bump into, and they do that similar to any type of hydrolysis that we've seen back all the way when we talked about digestion. They're just going to be ripping off single nucleotides from the end that they have access to, which is the 3' end. And so basically it puts a timer on the time that the mRNA can be functionally translated and turning into proteins. And so the poly A tail is creating basically a buffer so that mRNA has time to find some ribosomes and be translated into the protein product before it becomes so damaged that it can no longer produce viable proteins. So that's all part of homeostasis. We can't just let mRNAs persist forever inside of our cell, otherwise they would just constantly and endlessly be producing their products, and that's a homeostatic problem. Interestingly, it's also something to consider when we talk about things like the Pfizer and Mrna vaccines in terms of their ability to basically provide mRNA for your body to translate into the little proteins that we're trying to teach our immune system to recognize. They very quickly are going to be broken down inside of those cells. So there's a short duration during which mRNAs are functional before they are basically taken care of and broken down by the other protein machinery of the cell. That's a little bit about transcription. If you have questions, let me know. Next up is translation.